All right. Hello, everyone. So today I'm going to try recording myself giving a lecture, and we'll just see how it goes. It might go horribly. It might go okay. I don't know. This is just a test. First of all, I just want to say I hope everyone's doing all right. I know this is a very weird time. I myself am definitely figuring out how to adjust to this whole situation. How we're gonna, get some learning done, even though we aren't able to see each other in person. So we'll just try to get through it together. And the main thing I think is just to make sure that we're continuing to communicate, checking in with each other, and just, we just got to get through it together. But today I want to try to talk a little bit about protein synthesis. This is really supposed to follow up what we were working on before we left for the closure, the break. And so it is, really material that I was planning on covering. Um, in the interest of transparency, I'm not 100% sure if we're really supposed to be covering, I guess, quote unquote, typical curriculum. You might have seen some of the communication from the administration about, enrichment, and that is kind of the language that I've been using as well. But for those of you who are still interested in learning, the biology that we were planning on, I thought that I should still go ahead and have this available for you all. So let's try it out. I'm using an application that is just on my computer, so it may not be the best, but we'll just see how it goes. But anyways, protein synthesis, right? So synthesis means making things, right? I think we've talked about this word before. And so I talked about proteins and how they are actually really important in creating our traits, right? So we'll talk about how does it all come together today. And I'm going to first begin talking about something called RNA. Okay, so here's my slides, all right? So I think I put this in my previous lectures, but this is just showing the overall central dogma, right? The central dogma being how do we get from genes to traits, right? And so we have this in-between step of proteins. All right, and so protein synthesis is just how proteins are made using DNA as the instructions. And this specific example is talking about a protein called insulin. Okay, and so you might have heard of insulin before. People who have diabetes, they have issues with insulin, which basically helps you to process the sugar that's in your bloodstream. And so that's why sometimes people with diabetes, they may feel very faint, or, they have, issues with basically getting energy. And we'll talk hopefully more about that when we get to our fourth unit and how we process energy. Okay, so here again is our central dogma. So we start with DNA, and we've talked a little bit about it. And then so today I'm going to focus on this step, which is RNA. And you'll see there are some similarities between DNA and RNA, but ultimately it is an important intermediary step between DNA, protein, and then obviously we have kind of traits further down the line here. Okay, so what is RNA? So RNA stands for ribonucleic acid, and so you might think, oh, that's a bit of a familiar word. Well, DNA stands for deoxyribonucleic acid. So RNA, we've just dropped the deoxy, and it's just ribonucleic acid. And this is pretty representative of the differences between DNA and RNA. So you can see here in the picture, this would be a point in the lecture where I would maybe ask you to identify some differences. So I'll pause here and let you look at the two pictures of DNA and RNA and think about how are they different just to your eyes. All right, so what do you think? So the main things that I would point out here, and I think I may have some animations on these slides to show, but obviously the first thing that comes to my mind is that RNA is just a single strand, right? We talked about the structure of DNA. We've got this double helix, right? So it's a spiral involving two strands. RNA is still a spiral shape, okay, but it's just got the one strand. So that's one of the main differences. The other one that you might see is obviously in the name, right? We've dropped the deoxy in the deoxyribo. So that's RNA. And then the third difference that you might be able to point out is in this first base here, right? So we've got the letters of DNA, right? And you remember C pairs with G and T pairs with A. We have the same setup in RNA, but there is one major difference, which is instead of thymine, we have uracil. And so instead of T, we have U. So in RNA, C still pairs with G, but instead of T pairing with A, we've got U pairing with A. And this is mainly due to the structural difference in RNA. So you can imagine, right, it being single-stranded, it's the way that it kind of comes together is a little different. And so, uracil, if you look at the chemical structure of it, it's missing this little bit right here. And that is really the only difference between uracil and thymine. So here we've got our differences. Okay, so uracil replaces thymine, and it is single-stranded, right? And then you can see here the name is also different, and the name refers to the sugar, right? So if you remember back to our nucleotide components, there is a sugar involved. The sugar is slightly different. It's got a different name. Okay, so those are our differences. And here they are in a little chart for you, okay? So we actually have different types of RNA, and so these are going to be important because they have different jobs. So DNA, right, its main job, it stores the genetic information, right? It sits in the nucleus, and it has those instructions. It doesn't really do much else, okay? I mean, it's quite important, obviously, but its main job is to just store that information. But RNA is much more active, and it comes in different forms, and we will talk about each of these three when we get to their kind of place in the process. Okay, but we've got ribosomal RNA, okay, and we can shorten that, call it rRNA. And this forms the ribosomes. And so if you remember when we talked about cells, okay, hopefully you've done your cells review, the function of ribosomes is to make proteins, right? And so RNA is involved in just creating the structure of this organelle that makes the proteins. Okay, we've also got transfer RNA, which we can call tRNA, and it carries these amino acids. So if you remember, okay, this is again a bit of a review. Amino acids form proteins, okay, and so the tRNA is in charge of, carrying around these building blocks, all right? And then finally we have messenger RNA, and we will focus more on this today. So mRNA. Okay, it forms a message, and the ribosomes are going to use that as the direct instructions to create the protein. So it's like a little bit of a code that the RNA is carrying over to the ribosomes that they can use. Okay, and we'll see how does that work. Here are some pictures of the different types of RNAs. You can see that they've got different shapes, okay, and this is a bit of a theme that you'll see in this class, which is that structure informs function. So the way that things are shaped, it's because that they are doing a certain job, right? And so we've got messenger RNA is your typical, long strand. The ribosomal RNA kind of clumps together to form the ribosomes. And then the tRNA has a very interesting, what's called a clover leaf structure, and we will touch on that more later. So here again, this is a step in which we are at right now. And this step that we're going to dive more into right now is going from DNA to RNA. We call that transcription. Okay, so we have to create a code using RNA from the DNA. Okay, and so let's see how does that work, right? So why does the cell need RNA, right? So you might be thinking, okay, well, sure, the RNA is this code, this message that the ribosome is reading. Why can't we just use the DNA, right? Is that not enough? It's got the whole code in there. Okay, and that would be a fair question to ask. So let's take a look first at this picture. This is an image sort of zoomed in the cell. So you can see here, this is actually the nucleus. So here is the membrane of the nucleus. And as you know, our DNA is stored inside here. Okay, so it's sitting in here with our genetic information. However, the ribosomes, if you remember, they're out in the cytoplasm. They're outside of the nucleus. So some of them are floating around in the cytoplasm, and then some of them may be sitting on those endoplasmic reticulums, okay? And so we have a physical distance that we need to travel in order for the message stored in the DNA to get out to the ribosome. So let's think about for a second, okay, the DNA is double-stranded, and it's quite a long molecule, if you remember as well, right? One chromosome is one DNA molecule, and it can be millions or billions of base pairs long. So it's a lot, right? It's like a very large ball of yarn. If you ever seen those Guinness record balls of yarn or whatever it is. Okay, think along those lines. So it's not an easy task to be transferring the DNA in and out of the nucleus just for the code to get over there. Okay, and the other thing to think about as well is, the DNA, you don't want stuff to happen to it. So there's a reason why it's stored in the nucleus, it's so that it's protected. So we really want to keep it in the nucleus. But at the same time, we want the message to go out to the ribosome. So you might be seeing kind of where I'm going with this right now. But ultimately, right, the idea, let me go back. The idea is that the RNA is this simple way that we can take, bits of information that's stored in our DNA library and then transfer them out to the ribosome where they can be easily used to make proteins. Okay, and so one important thing to note is that the RNA is really the code that the ribosome is reading. It's not being directly used to create protein. It's like the instructions, right? The recipe book. So you can think of it as we have this giant recipe book inside the nucleus. We only need one recipe at a time, so we might just copy a page in the book and take it out, to the kitchen or whatever to make a protein. Okay, so that's why we have RNA. All right, another important reason that we have RNA is for something called gene regulation, okay? And so remember I was just talking about one DNA molecule, it's massive. It can have up to 3,000 genes, and each of those genes themselves are quite long. But you don't need all those genes to be turned on all the time, right? So some genes you might need in certain situations. Some genes might be turned off permanently depending on, what kind of trait it might be. Okay, you can think of, for example, our lactase gene that I've mentioned before, right? In most people, naturally, it turns off after you stop being a baby, essentially, right? So that might be a situation in which you would no longer need that gene to be expressed, right? And so if we have this intermediary step of using RNA, then it's easier for us to control which genes are being turned on at what time, right? So if we need this gene, then we can just use the RNA for that gene. But if this other gene needs to be turned off, then we'll just not even make the RNA for that gene. So having intermediary steps essentially gives you more control. That's the idea. Right, and again, here's my analogy of the recipe book. Okay, so we've got loads of recipes in there, but you don't need to make them all the time, right? You just need to eat one thing at a time. So we'll just take out one recipe, okay? All right, here is a comparison of the structure of RNA versus DNA. So really you can think of RNA as being just half of DNA, which also makes it a lot smaller. It's a lot easier to move around. It can go in and out of the nucleus as it pleases. All right, and so RNA is more mobile and it's also disposable. Okay, so we can make as many copies of the RNA as we'd like. And then because, it's actually quite dangerous in the cell sometimes for small molecules. After the RNA is used, it often gets degraded, right? So it might get picked up by those lysosomes, right, which are the vacuum cleaners of the cell, and it'll get recycled, and then we'll just make more of it when we need it. So it's, like when I make a copy out of a textbook or something for you, it's a similar idea. We can move it around and we can get rid of it when we need to. Okay, well, here's my reminder. Where are the ribosomes? Well, they're out in the cytoplasm. Okay, so just to sum it up, our cells need RNA because it regulates our genes and traits, and it acts as a disposable copy of the genetic instructions. Okay, and so the process in which we make RNA, right, again, it's called transcription, okay? When you transcribe something, normally it means you're hearing someone say it and then you write it down. Okay, and it's kind of a similar situation, right? So we have this information in DNA and we need to kind of write it down in the RNA so that we can use it. And this is going to happen in the nucleus, right? Because the DNA, you can think of it as being kind of trapped in there. Okay, so we make the RNA in the nucleus using the DNA as a template. And it's quite simple. Okay, it's very similar. Oh, let me go back here. It's very similar to how we make more DNA, right? So we just match up the correct letter. Okay, and then we just have to remember that instead of T's, we've got U's. You can see here they're red, okay? The U's are replacing the T's, and they match up with the A's, right? So U pairs with A pairs with T, C pairs with G, U again with A, G with C, okay? And then on, and you only have to make one strand, okay, because RNA is single-stranded. All right, and just to go back to our central dogma again, this is the step at which we're at, right? We're going from the DNA, we're making RNA through transcription, okay? And then we want to eventually get to proteins, and I will cover that in another lecture. Okay, here is another image of transcription. So you can see here we do have to unzip the DNA to get at it again, okay? And then instead of DNA polymerase, we have RNA polymerase, right? We have RNA polymerase to make RNA, okay? And then we are going to move in this 5' to 3' direction using this template strand, okay? And so the new RNA that you're making here, which is pink, right, it should match up to this template strand. But if you look at the code on the non-template strand, you'll see it actually should be identical, with the only exceptions of the T, right, and the U's should be exchanged, okay? Because if these two match and these two match, then these two must be the same. It's kind of like math. Okay, so you can go ahead. I will write this down, okay? So I'll give you a moment, and this is just a little bit of practice. So here we have our template strand of DNA, right? So what do you think our RNA should be? So I'll give you a moment. I would pause the video right now, okay? So I'd pause it here, write it down, and then I will give you the answer in a moment, okay? Okay, so I gave you a little bit of moment. Hopefully you did pause the video to write it down. Let's just go through this, right? So T in the DNA would be paired with an A in the RNA, okay? Same as with DNA. But then A then pairs with U, okay? I don't know if I actually have this. No, I don't. All right, let's go back. So the T pairs with the A, all right? And then the A pairs with the U, the C pairs with the G, okay? The G pairs with the C. The A pairs with the U again. Okay, so I will just read out, because I forgot to actually write it out for you. I will read out what you should have written for your RNA. So it should be A, U, G, C, U, A, G, U, C, C, A, G, U, C, G, G, A, U, C. Okay, so should be pretty straightforward. We're just doing the same thing that we were with DNA, except this time we are going to be replacing the T's with the U's. Okay, so the next step, okay, is translation, and we will save that for a later date. Okay, but hopefully that was understandable enough, despite the fact that I cannot see you and you cannot see me. But we are going to see if we can try to at least continue learning about genetics despite this challenging time. Okay, so I will see you in my next video. Okay, feeling very YouTube right now, but hopefully that was helpful, and please let me know if there are any issues or questions that you have. And again, we're gonna have to get through this together. All right, I miss you all very much. I hope you're doing well. I hope you and your loved ones are hanging in there. So I will see you again next time.